Good afternoon and welcome to the BBC. That's the Bacteria Broadcasting Corporation, where the news multiplies faster than E. coli on a warm day. Today we will dive into the big question: can new life be created? We'll be lighting up the room with jellyfish, doubling with cloning and poking around with stem cells. Stay tuned, things are about to get organically interesting.
Let's drift into the world of jellyfish. These remarkable creatures can repair damaged body parts and can even revert themselves back to earlier life stages. Here's the science. Jellyfish already store stem-like cells. When an injury happens, these cells migrate to the damaged site and form a bundle of undifferentiated cells and rebuild whatever is missing, like tiny biological construction workers with no union break. And then there's the immortal jellyfish, which can turn its adult cells back into unspecialized ones under the right conditions. Others skip regeneration altogether and simply rearrange what they already have left using muscle-driven forces. Efficient and lazy, a true inspiration.
Next up, stem cells, including embryonic stem cells and induced pluripotent stem cells. These are blank cells that can turn into muscle, nerve, skin, or organ tissue. Research mainly focuses on repairing and replacing tissues on a small scale, but if scaled up, entire organ systems could theoretically be made from one bundle of small cells. Stem cells naturally differentiate under specific conditions. Scientists can stimulate the differentiation using man-made cues like electric shock or by modulating the environment to mimic a room. Interestingly, stem cells behave differently in space. Orbit experiments show changes in gene expression due to altered gravity, accelerating development, and creating new genetic variation. This shows how stem cells could, in theory, contribute to creating new life or organs.
Of course, the ethical debate is unavoidable. Using embryonic stem cells destroys embryos, which is simply seen as morally wrong. Others argue the potential medical breakthroughs, such as creating new life, outweighs these concerns. It's a very delicate balance.
Now, on to cloning, creating a genetically identical copy of a cell, tissue, or entire organism. There are three main types: gene, reproductive, and therapeutic cloning, each with a different purpose. The most famous example, Dolly the sheep. Created in 1996 from an adult, Dolly lived a normal life until 6 years old, proving cloning works, but she had a short life and complicated health issues. Cloning can aid medical advancements like generating organs for transplant with reduced rejection, conservation, and even high-yield livestock. But cloning only copies existing DNA. It can't create a fully unique organism. Personality, upbringing, and experience are not cloned, only genetics.
So weighing the cons, moral concerns over embryos, playing God, unintended ecological harm, animal welfare, reduced biodiversity, and bioweapon potential. Synthetic life is powerful, but it can be misused.
Against all that, there are huge pros: medical breakthroughs, curing genetic diseases, environmental benefits like pollution cleanup, conservation of species, lab-grown tissues, reducing animal testing, and even new scientific understanding of new life.
So it all comes down to balance, using science responsibly, weighing risks, and rewarding and asking ourselves, how far should humans go in creating new life?
That's all for tonight on BBC, the Bacteria Broadcasting Corporation. We've explored jellyfish, stem cells, and cloning, and the ethics behind creating new life.
So, can new life be created? Our answer, yes, but not yet.
Science is making incredible strides, but fully creating new, unique life is still on the horizon.
Until then, we can keep learning from nature, pushing the boundaries, and exploring possibilities.
Stay curious and keep asking big questions.